Spectrophotometry. Spectrophotometry, AP Bio Jones. All right, spectrophotometry. This is a Spec 20, and we're going to measure some food dyes, actually. And we're going to use this to understand how we will measure our DPIP dye in our photosynthesis lab, which we're going to do later in the week. Okay, so this is just an introduction to spectrophotometry or spectrophotometry using a Spec 20. Here we go. This is an analog spec, mind you. This is not digital, it's analog. All right, so the first thing we do with a spec, or this spec in particular, because they're actually all different, is we zero it. So all I'm doing is bringing the dial to 0. I'm bringing the analog dial to 0. So I'm telling the machine to start at 0, which is a good thing. Then I'm going to put what's called my blank in. This is my blank. Notice I'm holding it from the top because I don't want to touch any of this. Any of this I don't want to touch, because if I get my fingerprints on this, not good. That will bend light. That'll diffract light, right? In fact, I'm just going to quickly wipe this off with a Kim wipe. I had a student named Richard Kim. I used to call Kim wipes. He loved it. We laughed and laughed and laughed. Anyway, this is the cuvette, special expensive glass that allows light to go through it, but not diffract or refract all that much. That's why it's so expensive. They're test tubes, but they're special test tubes. Okay, so I'm zeroed out. This is my blank. I'm going to cancel out everything in here. Here we go. I'm going to put the blank in the chamber, close the hatch, and then, if you notice, it goes to 88%. So that's 88% transmittance. In our lab in photosynthesis, we're going to use transmittance. That's percent transmittance. That's how much light can transmit through the solution. So that means 88% is transmitted through. That loosely means that 12% is absorbed. But for absorbance, we have to actually use a log scale. And for absorbance, it's actually 0.05 or almost 0.04 really. Or 0.06, excuse me. But that's a log scale of transmittance. We're not using absorbance, so I will not go over the specifics of absorbance. But think of absorbance as the flip-flop, the inverse of transmittance. This is 88% transmittance, okay? So loosely said, not accurately, you can say 12% is absorbed. But this is the blank. So I'm actually going to turn this knob, you can see it down here, to 100, because this is the most important part of the whole lab, arguably. So what I did was I put the blank in, and I told the machine that 100% of the light goes through that blank. The fix is in. It's an expression, meaning all the light can go through the glass in that water. It can't, but I'm telling the machine it can. So everything downstream of this cuvette is compared to these parameters. This is called a blank, because I'm blanking out the glass and the water and anything in this water. This is just tap water, but I'm canceling out everything in here. I'm canceling it all out. It's called the blank. All right, so now I take my first experimental cuvette. This is just food coloring. This was the blank. This is the experimental cuvette in the blue. The experimental cuvette is simply put right back in. Well, first I check if it's 0, and it is zeroed. It should be, because I zeroed the machine. I put the experimental cuvette in, and as you notice, the dial didn't go to 100. It went to 91-ish%. So what that means is that 91% of the light compared to the blank can transmit through the cuvette. And that's because I canceled out all of the light from the blank. So I let all the light go through, in other words. So 91% of the light is allowed to go through. So that means, roughly said, 11% is absorbed. It's not actually 11, but Don't worry about that. 91% is transmitted through compared to that blank. All right, so again, this is 100%. That's the blank. This is 91-ish percent. Was allowed to transmit. So this blue stuff in here is absorbing light, obviously, and not allowing 100% of light to transmit through. Here's my next experimental cuvette. This is a different one. I know they look the same, but it's a different concentration. It's a different concentration. So I'm going to put this one in, close the hatch, and this one goes to 97%. So obviously, if everything's the same, this is a lower concentration. It's a lower concentration. Concentration. This is where Beer's law comes in. This is where Beer's law comes in, which is actually not the scope of this lab. But if you take chem, this is where Beer's law would come in. For this lab, our concentrations are identical. So we're not using Beer's law. But if you were taking it, this is where you would bring it in, concentration. I just did different degrees of blue just for fun. Okay, so lastly, here's my blank. Here's the first one I put in. Here's the second one I put in. Here's the third one. You can see it's actually lighter blue, right? Now this one, I have over 100%. So I have 100+% transmittance. So that means something's up, all right? Now that has to do with the fact that I asked this class in particular, what wavelength do you want? And a student in this class said they just chose 500 something, 500. So I just chose 500 randomly, all right? But let's see any difference that we would get with a different wavelength. Blank. My glasses so I can see this dial. I'm gonna jump 100 to 605 nanometers. I just jumped 100 ahead. I'm going to zero out my spec. There we go. I'm zeroed. Whoops. Oh, hold on a second. Wait a tick. Wait a minute. Zero out. There we go. Zero out. Blank. Wow, it's actually absorbing a lot. That tap water has a lot of solutes in it. Dialing to 100, take out my blank, put my first experimental in. Look at that, 12%. What a difference, right? 12%. What a difference 100 nanometers in wavelength makes. Here's the next one, same order as before. Wow, 8%, 8%. Third one, whoa, 70%, right? So what we're seeing is that just by changing the wavelength, from 500, because we chose 500 randomly, to 600, we jumped up 600 into the reds, into red light. Just by changing it 100, we have very different values, right? Really different absorbances, and therefore transmittances, really different transmittances. So far less light went through. So that tells us that, take that out, that tells us that this dye is much better at absorbing higher wavelengths and much worse at absorbing lower wavelengths. And what color is this dye? Anybody want to volunteer?
Blue?
It's blue. And what color is 500-ish?
I think it's around green.
It's around blue-green. So it makes a lot of sense, right? It makes a lot of sense that 500 wavelengths of light was not absorbed greatly by this. Because this is already blue. It's already reflecting that light. So the molecules in this solution reflect bluish light, which is around 500 somewhere. This is actually green. It actually says green on the bottle. It's green food color. It just looks blue. So that's why, and again, that 500 was chosen randomly by a person in this class. Okay, she said 500. Okay, cool, but 500 on. So we put 500 on. So I was hoping they'd pick somewhere in the 500s because I wanted the light to go through this radically, and it went through this radically. But when we went to 600, it was really absorbed by this. So that tells us that this blue food coloring absorbs red light and doesn't absorb blue-green light. And that's obvious because it is blue-green, right? It is blue-green. My glove is blue because it's not absorbing blue light, right? It's reflecting bluish light at your eyes, at my eyes. That's how we know it's green. Blue, excuse me, blue. And this little red cap, this is red because it's absorbing other wavelengths than red. I don't know which ones they are, but they're not red. I know that much. I can see this, right? And the red light's bouncing off this and going into my eye, right? Sending a signal to my optic nerve. And then my brain communicates with the optic nerve, and I construct this color to be red. And I've learned that it is red. It is red. And I know that. I've learned that, right? If someone taught me that this color was elephant, I'd think it's elephant, but it would still appear red to me. I just wouldn't call it red.
What about colorblind people?
So, yes, colorblind people. There's different types of colorblindness, but generally it is a result of brain activity not distinguishing two wavelengths or more wavelengths and blending them into one. And that's the short answer of it. And we'll hit that again in genetics and sex-linked genetics, as colorblindness is. But pretty cool, right? So back to the spec, not to roll over that question, but back to the spec. Just to summarize, guys, I'm going to shut the video off. Just to summarize, this is blue-green. It looks blue, I know, but this is blue-green, and it did not absorb blue light, the 500. But once I dialed the spec to 600, it really absorbed it far better, right? Much more, like 80% more was absorbed, and therefore not transmitted. Because obviously, it's blue-green, and it doesn't absorb blue-green light. All right, that's the Spec 20. Let me turn the video off.